And
we're
live now. Yeah.
Okay. Welcome everyone to episode 5 of season 3 of the Future of Foraging seminar series. Today's episode is going to be about plant-animal interactions and specifically pollination, a curious case of cross-kingdom cooperation. Our two speakers are Michael Harrap and Anna Stöckl. And Michael will kick us off with her talk on overlooked floral display traits and their influence on pollinator behavior. So this first talk will cover, think more proximal cues, and then we'll have a second talk by Anna Stöckl on more than just a pretty flower, how visual patterns guide insect-plant interactions. And this will cover, think, visual cues and sort of longer range interactions between flowers and insects. As always, there's a little bit of time to ask questions after each of the talks. And even during the talk, you can use the Ask a Question box at the bottom of your screen to write out your questions. And other people can also vote for questions they think are interesting and would like to be asked to. And then after the two talks, we'll have a general panel discussion where we will cover sort of a broader questions and comments on the two talks. And with this, I'll hand it over to Shivam, who will introduce Michael.
All right. It's my pleasure to introduce our first speaker, Mike Harrap. Mike has looked at pollination from both the perspective of the pollinator as well as the pollinated. First, as a master's student at Durham University, studying the effect of flower shape on pollinator movement, and then as a PhD student at the University of Bristol, where she began studying the role of floral temperature and humidity in pollination. She then moved to Oxford as a postdoc, where she pursued these questions, and this is what we will hear about today from her. With that, Mike, the screen is all yours.
Okay. Okay, can I confirm that you can see my screen and pointer? Yes, excellent. So should I just start now? Yes, yes?
Yes.
Okay then. So hello, I'm Michael, and as discussed, we're going to talk about some work I've been doing from across my career, looking at the role of overlooked floral display traits in influencing pollinator foraging choices. Straight off the bat, I would like to thank my collaborators and supervisors across various institutions, as well as the funders for the research I'm going to talk about today. Okay then. So, flowering plants in a natural environment have a problem. And that problem is they are competing with all the other plants in their environment for the attention of pollinators. To get around this, flowering plants produce attractive floral displays, which are attractive to the pollinators, but can also be learnt and recognized by those pollinators, allowing those pollinators to focus their foraging effort on particular flower species, allowing a more consistent pollination service. Similarly, these foraging pollinators, which are often insects such as bees, have a problem as well. And that is that different flowering plants differ in the rewards they provide. Flowers can differ in the quantity, quality, and the actual type of rewards they provide, so like pollen and nectar. So in order to maximize foraging success, these foraging pollinators need to be able to recognize the most rewarding flowering plants in their environment and adjust their foraging behavior accordingly to favor those more rewarding flowers. To do this, as we said, similarly, they learn the floral displays that the flowers produce and use them to inform their foraging choices. So, floral displays are multimodal. They consist of many different signals and cues which pollinators can recognize, learn, and respond to. Each of these signals and cues being a signaling modality. So here on this diagram, we have a list of all the different floral signaling modalities that we know at least some pollinators can respond to. We also get patterns in floral signaling modalities, which is a change in the intensity or composition of a floral signaling modality across the flower surface. So looking at this list, we can see that there are some modalities that we as humans can notice, and these have been the focus of research in the past. We know that flowers are colourful, they produce visual modality cues, but they also smell, they produce olfactory cues. In addition to these cues we can detect, there are also cues that are less obvious to us as humans, who are not the target organisms, which pollinators can respond to and detect, but we can only detect using specialist equipment. So as we've said before, throughout my career, a big focus has been looking at the role of these overlooked floral signaling modalities and trying to perhaps readdress the balance of the research to try and get our understanding of those modalities up to a level that's comparable to the ones we can detect ourselves. So today I'm going to talk about some work I've done looking at floral temperature and floral humidity, which are two of these overlooked modalities. And we're going to start with work on floral humidity. So what is floral humidity? Well, the concept of floral humidity is quite straightforward. Many flowers contain liquid nectar, and compared to other plant surfaces, floral tissues tend to be quite leaky. They're permeable to water, and some flowering plant species even have stomatal pores on their petals. Through a combination of evaporation of liquid nectar and transpiration or water loss, many flowers create an area of elevated humidity around the head space of the flower, and this is the floral humidity. So floral humidity was first identified by this group at Cornell, who found the evening primrose Onyphera cespitosa produced floral humidity, and that the hawkmoth pollinator of this species had a preference for flowers producing floral humidity. However, liquid nectar is quite common in flowering plants, and transpiration is a ubiquitous plant process. So it makes sense for other flowering plants to be able to produce floral humidity. Additionally, hygroreceptors, humidity-sensitive cincilli, are really common across insects and are also found in generalist pollinators such as bees. So what we wanted to find out is whether floral humidity occurs in other species and whether it can be used more widely by generalist pollinators outside of this specific hawkmoth primrose pollination system. So for the first part of that, working out whether other flowers produce floral humidity, we used this robot. So to this robot, we got flowers and put them in a water-filled and sealed vial and presented them to the robot. This robot has a humidity probe on a long arm, and it moves this humidity probe through the flower's headspace in a set transect sequence, measuring humidity at specific points relative to the flower as it goes. And this sequence looks something like this. So it's worth saying, in this video, the robot is moving at about 3 times the speed it would normally, and it's not actually stopping to take measurements. So the real sampling would be happening at a considerably slower speed. This is just to show you the layout of the transect. So using this method, we sampled 42 flower species, including resampling the evening primrose Onyphera cespitosa. In addition to this, we sampled 6 controls, all of which were variants on the water-filled and capped tube that we presented flowers to the robot in, just without a flower present. So this transect method does produce structural data, so the changes in humidity across the transect as we go through the flower's headspace. But on the whole, floral humidity is greatest at the center of the transect, where it's nearer the center of the flower. So what I'm actually going to present to you today is one of our summary values. This is delta RH max. So this can be thought of as the average peak difference in humidity relative to the background produced by each flower species or each control. So here we have our 42 flower species, and our 6 controls. The flower species are in blue, the controls are in orange. The important controls are these two. The water lid control on the left is the water-filled and capped tube without a flower present. This is basically the tube as it was when we presented flowers to the robot, just without a flower there. The other important control is this one on the right, the water control. This is that same tube with the lid actually removed. So this can be thought of as the most humidity the water present in the tube could have produced if it were unobstructed. So looking at this data, we see that floor humidity varies between species. You have some species that produce very little floor humidity. You have some species that produce an intermediate amount of humidity. And some indeed produce quite a lot of floor humidity. Within those species that produce quite a lot of floor humidity, you do have the evening primrose, Onotherus espitosa. But not all of these species that produce a lot of humidity are pollinated by hawkmoths. And it includes species that might be visited by generalist pollinators, such as bees, like California poppy, bindweeds, dandelions. So floor humidity varies, and it doesn't seem to be limited to moth-pollinated plants. So we know there's variation in floor humidity on flowers that bees might visit. So now we need to find out whether generalist pollinators, such as bees, can make use of floor humidity. And to do that, we use these captive bumblebees in these flight arenas. And, excuse me, and we see whether bees can respond to artificial flowers that produce a floor humidity cue comparable to that seen in nature. The reason we use artificial flowers is that real flowers, being multimodal, will produce many different kinds, different modalities of cue, and it will be very difficult to find a real flower that differs only in floor humidity. So we use artificial flowers where we can control the cues that we present to the pollinators, and we can also control the reward state of those flowers more carefully. So the artificial flowers themselves for this experiment, there are 2 variants. There's a humid variant of the artificial flower that produces an area of elevated humidity in the headspace of the flower, and a dry variant that produces less humidity, but otherwise the flowers do not vary. I'm going to show you data today from experiments using these artificial flowers. These artificial flowers have a permeable top, and inside them there are sponge components. For the humid flower variant, we wet these sponge components, and that allows the flower to create an area of elevated humidity in the flower's headspace, floral humidity, by evaporation of water through the permeable lid on the top of the flower. For the dry variant, these sponge components are put in dry, so they don't produce as much humidity. As you can see on the right here, these flowers also have a little feeding well, which we can put food in the flowers for the bees, so we can control the flower's reward state as well. So when you sample these flowers through the same humidity sampling procedure I described before, you see that they produce a level of floor humidity comparable to that seen in nature, with the humid variant flowers producing a level of humidity comparable to the highest humidity producing species, and the dry variant flowers producing something more comparable to the intermediate humidity producing species. So it's a biologically relevant humidity cue that we're testing the bees' response to. So to see whether bees can use floor humidity, we're going to use a differential conditioning protocol. I'm going to talk you through this because we're going to use this a few times in the talk. So we present individual bees with several artificial flowers, half of which are rewarding, half of which are non-rewarding, and we allow individual bees to forage freely on these flowers for several visits. We monitor the bees' landing and feeding decisions and use that to evaluate the bees' success at learning the reward state of flowers. This evaluation of success is looking at the proportions of correct visits made by the bees with regards to the reward state of the flower. So a correct visit would be either landing on a rewarding flower and subsequently drinking, or landing on a non-rewarding flower and departing without drinking, as both those actions are the correct thing to do given the reward state of the flower the bee is visiting. An increase in proportions of correct visits indicates that the bee is identifying the reward state of the flower and responding accordingly. So it's learning. So by varying the cues associated with rewarding and non-rewarding flowers, we can see what difference the addition of a cue that we're interested in, like humidity in this case, has on the bee's ability to learn which flowers are rewarding and which are not. So we have 3 conditioning groups. We have a control group where rewarding and non-rewarding flowers do not differ in floral humidity, and 2 conditioning groups where they do differ in floral humidity. We just change whether that elevated humidity cue is associated with rewards or a lack of rewards. So by comparing the bee's learning in these conditioning groups against the control, we can see what effect a humidity difference has on the bee's ability to learn floral reward state. So, get on to some results. We allow the bees to forage for 70 visits, and we monitor the proportion of correct visits made at 10 visit intervals. So, when rewarding and non-rewarding flowers do not differ in floral humidity, the bee shows a limited improvement in foraging success with experience. However, when rewarding and non-rewarding flowers differ in floral humidity, regardless of whether that elevated humidity cue is associated with rewarding or non-rewarding flowers, that difference allows bees to improve in foraging success with experience and show a higher level of success overall. So this is showing that bees can detect these biologically relevant floral humidity differences and then use that information to make learned foraging choices and adjust their foraging accordingly. So what we have effectively done is expanded our understanding of floral multimodality by adding this clarification to the list. Basically, floral humidity is not limited to that evening primrose hawkmoth system. It occurs more widely and can be used by widespread generalist pollinators such as bumblebees. So, with that said, let's talk about floral temperature. So, flowers, when they're outside, they tend to be hotter than their surrounding environment. This tends to be due to interception of solar radiation, which is a fancy way of saying they warm up in the sun. And you can tell this using thermal imaging. As you can see in these thermal images, the red half of the image is the hotter half of the image, and the flowers are comfortably within the red. So different flower species can differ in their capacity to warm up in the sun, and this can result in different flower species achieving different temperatures. It's been found that various bee species can respond to these differences in temperature, and bees can distinguish flowers as long as they're at least 2 degrees centigrade different. However, when you point a more modern thermal camera at flowers, you see that they don't heat up evenly. There's a difference in the intensity of temperature across the flower surface, so a temperature pattern. Frequently, the center of the flower tends to be hotter than its periphery, as seen in this daisy, the California poppy, or the rock rose. But in some species, that pattern is reversed, with the periphery being hotter and the center being colder, as in the hydrangea and this periwinkle. And in some species, it's specific structures that heat up, such as the landing pad of this crinum species. So we wanted to look further into floral temperature and see whether bees can actually use the arrangement of temperature within flowers, the temperature pattern, to inform foraging choices. So firstly, we wanted to get an idea of how common temperature patterns are. So we sampled a little over 100 species in roughly comparable conditions and looked at the temperature contrast within these flowers. So this is the difference between the hottest and coldest part of the flower. And as you can see in this crude diagram, a little over half the species we sampled had a temperature contrast that was at least that 2 degrees centigrade that we know bees can distinguish separate flowers by. So it seems that it's quite common to have a contrasting temperature pattern within flowers. So having established that, we are going to reuse that conditioning protocol I described before, but this time the flowers will differ in floral temperature pattern arrangement as opposed to floral humidity. So we have two temperature patterns that we create, a circle pattern and a bar pattern. And these are created by putting heating elements under the surface of these artificial flowers and changing the arrangement of those components to create different temperature patterns. Because the same components are used, the actual amount of heating that happens doesn't change, just the arrangement of that heating. So, using the same conditioning protocol here with floral temperature patterns, when rewarding and non-rewarding flowers do not differ in floral temperature patterns, effectively when we turn off the heating elements, bees show a limited improvement in foraging success. In fact, here they continue to forage pretty much randomly throughout the experiment. Remember that half the flowers are rewarding, half the flowers are not. So a randomly foraging bee would have a 50-50 chance of getting it right. However, when rewarding and non-rewarding flowers differ in floral temperature patterns, regardless of whether that pattern is associated with the circle, regardless of whether rewards are associated with the circle pattern or the bar pattern, bees show an improvement in foraging success with experience and reach a higher level of success overall. So this is again, as before, showing that bees can detect these temperature patterns and use them to make informed foraging choices. The presence of a temperature pattern improves the bee's ability to learn which flowers are rewarding. So as before, we've expanded our understanding of floral temperature patterns and floral multimodality more generally by basically adding patterns to the list of floral signaling modalities. So up to this point, I've been talking a lot about expanding our understanding of floral signaling modalities, but that's basically been limited to making the list longer, seeing what traits can be detected and have been overlooked floral signaling modalities. For the rest of this talk, I'm going to talk a little bit about some work I've been doing looking at fitting these overlooked modalities into the wider picture and seeing how they function alongside other, perhaps more established floral cues, such as visual cues. So there are many potential benefits from presenting a multimodal display as opposed to a unimodal display. That is, presenting lots of cues at once as opposed to just one. And these can be separated into three different groups, but these categories are not exclusive of each other. It's been suggested that presenting multiple cues enhances the pollinator's responses by allowing the flower to present more information or be more distinctive from other flowers. It's also been found that modalities can interact with each other to potentially enhance the pollinator response. It's also possible that different modalities may have different roles within the floral display, such as different cues signaling at different ranges, or some modalities perhaps indicating reward location, others indicating floral identity. It's also been suggested that some modalities may have a role in providing context. So the presence of one cue primes the bee to look at the others or attend to the others. And lastly, signaling across many modalities may allow the floral display to be more robust to certain modalities being disrupted, for example, by weather conditions. So humidity and scent might be affected by wind conditions, but color is less likely to be affected by the wind. So we wanted to look into this a little more, and the first thing we looked at was cross-modal transfer of pattern learning. So this is a form of signal interaction where the idea is learning of a pattern in one modality primes the bee to recognize that same pattern in another. And this was first identified by my colleague David Lawson, who found that when you condition bees to a scent pattern, they respond preferentially to a matching visual pattern. This may allow the bees' pattern learning to continue independently of that scent pattern being disrupted, for example, by wind. So it would be beneficial to both the plant and pollinator to maintain that learning. So we wanted to look into this with regards to floral temperature and visual patterns, because dark colored visual patterns often result in large amounts of heating. So matching visual and temperature patterns occur quite frequently. So to look into this, we did a sequence of tests using similar artificial flowers to what I described before, temperature pattern flowers and flowers with matching visual patterns. And we did a sequence of tests where we trained the bees on one modality and then tested their response to the new modality that is matching in patterns, but the bee has no prior experience of. And we did this both ways, so conditioning them to temperature patterns, seeing how they respond to visual patterns, and vice versa. So what you're going to see in the graphs in the subsequent slide are the success at the modality we conditioned bees to, and their subsequent response to the second modality. The colors correspond to the same sort of test groups as the previously described temperature pattern experiment, with a control where bees received no conditioning, and two groups where they were conditioned to one other. I'm sorry, it's gone dark for me, just technical difficulties. I'm sorry, something's gone wrong with my computer. Can someone confirm that I'm back, please?
We can hear you. The screen is still not yet. We don't see your slides. It should be fine. I don't think.
I'm sorry, my computer just went black for a second.
Totally
Can you
fine.
see me now?
Yes, now the screen is back, the PowerPoint presentation is back, and we can hear you. So
are we back where we were? I'm sorry.
Yeah, exactly.
I think I had an internet hiccup and my computer didn't like it. So we're good to go? Good to continue?
Awesome.
Okay, sorry about that, everyone. So as I said, you're going to see the conditioning in the first modality and the response in the second one. And the colors of the points respond to the test groups, which are the same as in the previously described temperature pattern conditioning experiment. Okay, sorry. So when we actually did this sequence of experiments, we found that the bees' response to the first modality had no bearing on their response to the second. So bees were more than capable of learning a temperature pattern, but that didn't affect their response to a visual pattern, and the same was true in the other way. So conditioning to a visual pattern did not affect the bees' ability to respond to a matching temperature pattern. So this would suggest that this kind of cross-modal transfer of pattern learning does not occur between these two modalities. So moving on, we wanted to look at floral guides. So a floral guide is a pattern of a floral signaling modality that corresponds with the reward location in the flower. This helps improve the flower handling in terms of speed and accuracy of the pollinators when they're visiting the flowers. This, by reducing time and effort costs, can make the flowers effectively more rewarding to the pollinator, and also can allow the plant to pilot the pollinator to where it wants to be and approach it in the correct way. So visual guides, sometimes called nectar guides, are quite well established and are quite well studied. But additional modalities like texture and scent have also been found to work as floral guides when those patterns correspond with the reward location. So when you combine different modalities together in a pattern that corresponds with reward location, it has been found that the guide functionality of these patterns is not improved by additional modalities. That is, when visual patterns and scent patterns correspond with reward location, flower handling and accuracy hasn't been improved beyond that of the best performing pattern that makes up that multimodal pattern. So in this case, that would be the visual pattern. So we wanted to look at whether temperature patterns on their own can function as a floral guide, much like visual, scent, and texture, because they often correspond with reward location. And we also wanted to see whether adding temperature patterns to a visual guide, as they often correspond with these dark patches that function as visual guides, results in enhancement of the guided response beyond that of the unimodal components. So to do that, we use these artificial flowers, which have two feeders, two of which are non-rewarding, one of which is rewarding. To some bees, we presented flowers which had no guides indicating reward location. To others, they had a visual guide indicating reward location, and others had a temperature guide, a temperature pattern indicating reward locations. This could either be a very hot pattern or warm pattern, which was slightly less intense. And we had an additional group where we presented both the visual pattern and the temperature pattern, the warm temperature pattern, together. So we filmed the bees' responses to these flowers. Sorry, that jumped. To these flowers, and monitored several metrics of handling that have been associated with floral guides. So this is a bee very early in the experiment. This particular bee has the rewards indicated by a temperature pattern. They're here. But this bee doesn't know what to do. It managed to leave without finding rewards. This is the same bee later in the experiment when it's actually started to learn what to do. And you see that time it found the rewards right away. So that's a little bit of a spoiler for what is going to happen. But yeah, so we looked at several floral handling metrics associated with handling speed and handling accuracy. We found that speed wasn't affected by the presence of temperature patterns, but accuracy was. And I'm going to show you one of those metrics now, looking at handling accuracy. So this is first feeder visits, the proportion of visits where the bee first attempted to feed from the rewarding flower. This is the proportion of visits where the bee's first attempt to feed was from the rewarding feeder. So when the bee hasn't got a guide to indicate reward location, it has a 1 in 3 chance of getting it right. When the bee has a temperature pattern, if it's not very hot, they have a slightly improved initial success, but improve with experience. With the hot guide, you still get that improvement in experience, but actually the initial success isn't as much. And we think that's because the hot guide is actually quite off-putting for the bees. But what this shows is that bees can be guided and learn to use thermal guides to improve their handling of flowers. So temperature guides do occur. When we gave them a visual guide, you saw both of these things just enhanced further, a greater level of initial success and greater learning with experience. So this is nothing new. This is a well understood how visual guides work. But when you combine temperature guides and visual guides together, these show an improvement in foraging success with experience and reach a higher level of success earlier in the experiment. So this is showing a multimodal combination of, this is showing when you combine temperature patterns and visual patterns together, the bee's response is enhanced beyond that of the unimodal components. So I've talked a lot about how these temperature patterns might function or might not alongside visual cues. Exploring all the different ways the response can be enhanced is an ongoing amount of work being conducted by many people. But I'm going to finish today with this particular point. When you combine visual cues with temperature cues, this gives a contrasting outcome to work conducted by my colleague looking at combining visual and scent cues together. And I've already discussed this. When you combine visual and scent patterns together, you do get cross-modal transfer of pattern learning, but not an enhancement of floral guide functions. But visual and temperature patterns, when they're combined together, you don't get cross-modal transfer, and you do get enhancement of floral guide function. So there's potentially many explanations as to why this might be, but the thought I'm going to finish on is that this demonstrates the need to consider these overlooked floral signaling modalities, because when you combine one modality with another, you get different enhancements to the pollinator's response and different outcomes when you combine that same modality with a different one. So in order to get a fully holistic understanding of what is guiding pollinator foraging choices, we need to consider all of these cues together, not just the ones that we can see ourselves. And with that, I think I'm going to finish. I'm sorry about the technical difficulties there. I overran a little bit, it seems. Okay, thank you.
That was great. I'm going to invite Anna and Laura and Hannah back on the screen.
Okay. So in the meantime, I'm going to ask, we don't have a lot of time right now, so we got a lot of questions. No, don't worry, but you know, it's like we got a lot of questions, so we'll leave most of them for later. I think they'll make good discussion questions anyway. But I think there was one from Laura that is more of an understanding question of your behavioral essay. And so I'll read out the question. So is this increase in success, which I think refers to the your measure of success in the behavioral essay, through learning because bees are more likely to feed when reward is there, or because they are more likely to leave when reward is not there? And let's see, so the comment is on this note, or if I misunderstood, but it is that, but is it that the bees learn about whether to feed if reward is available or not after visiting a flower, or whether they choose to visit in the first place? So I think, just to give a bit of things, I think
I think I do understand.
Learn, yeah, exactly. If you can.
So with the learning assays, if the bee just learns to try and feed on everything, it would be effectively just as likely to do the correct and the wrong thing. So with regards to what actually contributes to success overall, it's a combination of both landing and subsequent feeding choices. So the bee could choose to land on the rewarding flowers more frequently and drink every time, but it can only do that if it can detect the cue at distance. It doesn't quite make that distinction in terms of evaluating success. Whether the bee is avoiding feeding on the non-rewarding ones or visiting the rewarding flowers more frequently. Does that sort of answer?
For me, I don't know. Maybe Laura can ask later again.
I hope I've understood what's being asked.
Yeah. Don't worry. So we'll cover otherwise
Sorry.
again later. In the interest of time, I think we should move to a second speaker. Thanks again, Mike, and welcome, Anna. So it's a pleasure to introduce Anna Stöckl. Anna studied biology at Heidelberg, followed by a specialization in systemic neuroscience at the University of Munich. And she then moved to Lund to work on insect vision for her PhD, where under the supervision of Eric Warren, she worked on neural adaptations to vision in dim light in hawkmoths. And for her outstanding PhD work, she earned Best PhD Thesis Award from the university and a Young Investigator Awards from the Society of Neuroethology and the Society of Experimental Biology. After her PhD, she went to, I don't know if I pronounce this right, Aalto University in Finland for a brief postdoc on neural population coding in a mouse retina, and then moved back to Germany to start investigating spatial properties of insect vision. Initially as a research fellow at the University of Würzburg, and since June 2022, Anna is an eminenta group leader at the Department of Biology at Constance University, where her group uses moth as a model to study how insects process visual information to guide behavior. And I think Anna is also very passionate about science communication and has won several awards in this space. So I'm really looking forward to the talk today.
Thank you so much. You set the bar very high. I'll try to conform.
That was the plan. Now you're
now I'm sweating.
No, we good. All right.
Brilliant. Thank you so much for this invitation. I am going to share. I'm going to try to focus on my talk because I must admit I have so many questions already from Mike's wonderful talk but I'll try to focus on what we're talking about now. I hope you can see my screen.
Yes, we can see.
Yes, good. Thank you. Wonderful. Yeah. And I should say this again. Thank you, Mike, for introducing the topic that we're actually going to stay in. Pollinator plant interactions, and thank you for making my life a lot easier on jumping into this talk. I wanted to just take like a minute. Let me see if I can advance my slides as well. Here we go. To kind of set up how I come to this topic and to the questions of how insects interact with flowers and what cues they're using to do so. Because my interest in this has really started as Hannah has mentioned. I'm coming from visual processing, visual neuroethology in animals. So I'm interested in what information animals, and particularly in this case insects, pick up from the world, how they process this information through their eyes and then in their nervous system, and finally use this information to guide behavior. And I've been interested in different forms of behavior, many different types of locomotion control. And the animals we work with, hawkmoths and lately bees, they locomote often towards something to eat, and that is flowers. So in actually many different ways in various projects, flowers and interaction with flowers, attraction to flowers has sneaked in so that we finally also made this a bit more of a focus of our work. And again, it has been beautifully introduced that flowers are essentially the billboards of plants attracting pollinators to come, and that they actually use a multitude of different sensory cues, not just the obvious ones, which are the visual ones that I highlighted here. And full disclosure at the beginning, I'm working on the obvious flower cues, visual cues, and particularly visual markings on flowers that as early as actually in the late 18th century, a German naturalist, Christian Konrad Sprengel, has suggested that these markings on flowers can help pollinators be more efficient in their interaction with finding the nectary, potentially also finding pollen. And this idea has really led to the concept of what we've also heard about now, the concept of nectar guides or pollen guides, floral guides in general, where these contrasting patterns on flowers can help insect pollinators be more efficient. And it has been shown in a variety of species, of insect species, from more generalist foragers like bees, but also more specialized foragers like the hawkmoths, that indeed, if they are presented, at least mainly in a lab context, in a few studies also in the wild, with patterned flowers versus unpatterned flowers, that they are more efficient in their foraging, that they can find the nectar faster, essentially. Excuse me. And while this general benefit of having these floral markings or nectar guides presented on flowers has been very well demonstrated, it's not entirely clear at what stages of flower interaction these patterns actually play a role. And this was something that we wanted to investigate a bit closer. And we actually started not with our normal model species with hawkmoths, but we started this project with bees in a collaboration with Johannes Späte at Würzburg University, where we wanted to understand kind of all stages of flower interactions, from the approach to the landing on the flower to finding the nectary, and understand what roles flower patterns, visual flower patterns, I should say, actually play in this interaction. And kind of two main, I would say, hypotheses were our starting points on what these flower patterns might actually do. They might indicate the target, if you want, the target being the nectary, so showing the animals where to get to, ideally land most closest to be most efficient to find the nectar. Or they might be more like essentially like a hiking path sign or a sign to the buffet to indicate which trails, if you want, the animals should follow to get to the nectary on the fastest path. And so we looked at this in, as I said, in bumblebees, and this is mainly work by two students, Alexander Dietz and Robin Richter, who filmed bumblebees in a flight arena, not too different from the ones you've already seen, in their approach, as you saw, as they're interacting on the flower and as they're departing. And we essentially split the entire investigation of this interaction of the bumblebees with the flowers into three parts: the approach flight, and then the landing, and the time when the animals are actually on the flower and walking towards the nectary guidance phase. And as we then later realized, also a departure phase, as the animals are walking on the flower before they fly off. And what we could show, just to start with, oh yeah, and we were tracking the animals in this entire time so that we can then look specifically, how are they oriented, how are they moving as they're interacting with the flowers. And what we could show to start with is that indeed, as many studies before have shown, if we give the animals patterns on these artificial flowers, excuse me, particularly radial patterns that indicate also flower center that extend to the edge of the flowers, animals actually had a reduced handling time. And by handling time, I mean the entire time that animals started to approach the flower, which is an 8 centimeter circle around the flower center, from this starting point to actually finding the nectary, this entire time was 30% reduced if the flowers actually had patterns on them than if there were no patterns on the flowers. So this is just the onset kind of. We also saw in our study that if we have these visual flower patterns, very simplified, very abstract patterns, the animals were faster in their handling of the flowers than when there were no patterns present. And 30% faster, which is quite substantial. If we consider that a bumblebee might be visiting hundreds of flowers a day, it can visit considerably more if it's 30% faster. All right, so now we come to what stages of this flower interaction were actually affected by the flower patterns. And we first looked at the approach flight of the bumblebees, and actually here I show you just a very simple metric, and that is the alignment of the bumblebee's body relative to the flower patterns that we presented to them in the arena. And what you already see here is that I underlaid kind of the pattern in this distribution. You see that there was a higher proportion of animals that oriented their body aligned with the pattern axes as they were approaching the flower when we had such circular flower patterns, compared to when we had no flower patterns, because then they had nothing to align to. Then they would align in all axes relative to the center. And so this suggests that already as the animals are approaching the flower patterns have a function, and that is to help the animal essentially choose its flight path towards the flower and align their bodies, potentially optimal for then the next step, which is the landing procedure. And so what we did as a next step is to look at where did the animals actually land on the flowers. And I showed you here all landings essentially for our radial flower type again, where each little filled marker connected to an open marker is one bumblebee, and it's the head and the thorax of a bumblebee. And what you can hopefully already see in this general distribution is that the animals landed preferably at the points where the flower patterns intersected with the flower's edge. And they also landed more generally around the edges than at the center. Excuse me. All right. And what that shows us, well, it shows us many, many different points. There's quite a lot of information in this data, but what it shows us, first of all, is even if we have radial flower patterns that indicate the center of the flower, and it's also the place where the animals were fed before we were starting to let them fly to these patterned flowers, we gave them gray, unfeatured flowers. They were always fed in the center. Our radial patterns indicate the flower center, and yet the animals choose to land preferably on the edges, suggesting that even if they have a radial pattern, it's actually not pointing them to the center. It's not making them land where they should expect the nectary, but it's indicating to them maybe very helpful and clearly featured landing spots. And we could further look into this by using different types of radial patterns. So we had a cross pattern where the arms extend to the edge of the flower, and then a cross pattern where the arms essentially stopped halfway to the edge of the flower, and finally a radial pattern with only the outer portions of this cross. And what we saw is in all cases, the animals preferably landed, maybe you see my mouse, I just tried, preferably landed at the outer part of the flower. So this is the distance from the center in all three conditions. But there was a slight trend when we looked at the population analysis that animals tended to land, or a higher proportion of animals tended towards land towards the center. This is the orange colored bar, when we had patterns that actually ended further in to the flower center. So in this pink pattern case. So what type of flower pattern you present to the animals can shift where they choose to land. And the bottom line is they don't land where the pattern arms might indicate the center of the flower, where they might expect the nectary, but they land essentially at high contrast areas that the patterns provide. And they might do that because it provides a very safe or high featured landing spot for them. They might need this visual information to guide their landing. All right. And the next stage we could look at is how are the animals then approaching the flower center? Are they, for example, following the markings to the flower center? Are they faster from landing to getting to the nectary if we present them with flower patterns versus no patterns? And well, I give you a very short answer on are they following the markings. I did a lot of different types of track analysis, and I found no correlation between how the animals walked on the flower and where the flower patterns were or how they were oriented. And we also saw no difference in the time the animals took from landing to finding the nectar with the different types of patterns we looked at, also with, I haven't added them here, but also with the different, the shorter cross that only is in the inner part of the flower and the cross bars on the outside of the flower. So this suggests that at least in our experiment, and I should say this, where the bees had experienced artificial feeders before, but not feeders with patterns on them, the flower patterns made no difference to how fast the animals actually found the nectary once they landed. So they seem to make no difference on the actual nectary guidance on the flower. Weirdly enough, and I'm still, or we are all still a bit puzzled by what that means, they did make a difference, so the flower patterns did make a difference on the time the animals took to depart. So animals generally spend more time on the flower after they actually drank their nectar reward and walked a little bit around, also walked on, I can go back here, on much more wriggly paths than for their approach, which were very straightforward, the approaches. And interestingly enough, they departed the fastest when flower patterns or radial patterns were present, and the slowest when there were no patterns present, which is really something, yeah, that we don't have a very good explanation for, but I'm very happy to discuss what this might be. So if we summarize functions of visual, I should say again, nectar guides for bumblebees in our study, looking at all the different stages of flower interactions, what we found is that, at least judging from where the bees landed, the flower patterns didn't indicate nectary location, or we could also say it in another way, they didn't make the bees land where the presumed nectary would be. They also, or the bees did also not use these flower patterns to actually kind of walk along like guides to the nectary, and it didn't improve their nectary recovery time after landing. But what we could see is that these flower patterns had a very important function already in the approach flight of the bees, and their landing control. It's kind of more, if you want, locomotor functions or flight control and landing control functions that have nothing directly to do with the actual nectar recovery, but are very, very important for the bees to actually handle flowers in general. And then our interesting finding that they also impacted departure guidance. And so this is kind of the short gist of what we found in our study on how visual flower patterns guide the different stages of flower interactions of a generalized pollinator and a pollinator that is landing on flowers, as most pollinators do, in this case, a bumblebee. But well, because we have them in the lab and they're actually our main model animal, we also wanted to know how does this pan out for a very specialized pollinator that doesn't land on flowers. Oh no, and now I managed, let me just, I think you might be hearing, I don't know if you're hearing the sound, but I might be, can someone give me feedback if you're hearing the sound in this video?
We don't hear this.
Okay, good. Then I'm playing it. Then it's just annoying to me.
Okay. Sorry.
Cool. So yeah, so we were interested in how these, how hawkmoths, and we've already briefly heard about hawkmoths. I play it again. How hawkmoths might use flower patterns, as they are not landing on flowers. They're sticking their very long proboscis into the center of the flower, or tapping on it in search of the nectary. And if you think about it, this is also a really advanced motor control task, because the proboscis of this hawkmoth is as long as the entire animal. And while it's hovering in front of a potentially moving flower, it has to stick its proboscis into the nectary. And we were wondering what functions these visual flower patterns play in this task, and how that compares to what we found in bumblebees. So I'll walk you through some of the key findings and directly like compare of what we found before. So first of all, do the hawkmoths also use these flower patterns for approach orientation? So for angling kind of their body in relation to the flower as they're approaching the flower. And the very short answer is yes. So if we analyzed from flight tracks of the hawkmoths around these flowers, how their body was angled, we found in this case with just a simple line pattern where we can very nicely see if their body is angled towards it, that body orientation very much matched the main pattern axis in these hawkmoths. And we can also do this with different types of patterns. They have a very, very strong tendency, much stronger than in bumblebees actually, to align their body with the dominant pattern axis. And we can play around with this and, for example, shorten the pattern axis so that the ratio or the dominant direction of the pattern isn't so obvious anymore. And down to patterns that are 5 times longer than they're wide, we still see a trend in the orientation of the animal's body angle with the pattern angle. And as the patterns get shorter or actually symmetrical, we don't see this anymore. We see just like orientations of the animals in all axes around the patterns. But bottom line is this approach orientation of hawkmoths that is induced by visual patterns is very, very strong. All right, where do the hawkmoths? Well, the hawkmoths do not land on flowers, but if we consider their proboscis position, where they first contact the flower with their proboscis as their landing location, homologous to how the bee would actually land on the flower, we can have a look if they also use these visual patterns for that. And again, the answer is very much absolutely and utterly yes. So if we, again, in this line pattern condition, but we also looked at different types of pattern, and it's the same throughout these types of patterns. If they extend to the edge of the flower, the animal's first contact with his proboscis, and you see this here in the color indicators. If it's brighter or yellower, then you have more proboscis contacts. They are very much aligned with where the patterns are on the flower. So flower patterns or visual patterns extend to the edge of a flower. That's where the hawkmoth's proboscis will contact the flower first. And after landing, after contacting the flower, how do the animals then move their proboscis onto the flower surface? Are they using visual patterns for that? Unlike the bumblebees, which at least in our experiments didn't seem to care about the visual patterns on the flowers. So what about nectary guidance, if you want, on the flower? And what you already very nicely see in this video, if you follow the proboscis of the hawkmoth that we tracked here as they're interacting with these flowers, and I should say it's filmed in black and white, but we generally show the animals yellow flowers on a blue background. You already see, I think, very clearly that the animals are moving their proboscis along this visual pattern on the flower. And I should say all the flowers we tested were paper printouts and then laminated. So the surfaces of all of these different types of flowers, whether they had patterns or not, were the same. So there were no differences in mechanosensory cues. We can play around with this, and this is students in my lab have done this, Alexa Hahn and Nadine Kraft, with many different types of patterns. Here just 2 plus control that doesn't have a visual pattern. And if we track where the animals are actually placing their proboscis contacts, you see that they're very much oriented along these visual features of patterns. Whereas if we don't have a pattern on the flower, they are homogeneously searching for the nectary across the entire flower. And we could even go one step further and determine, do the animals not just they might use different mechanisms to actually move their proboscis along the flower patterns. And we wanted to know, do they actually need to see or get visual feedback for this behavior? Do they need to see where they place their proboscis and see kind of the flower patterns in their frontal visual field when they're interacting with these visual patterns? So is this really a visual behavior? And to do so, my master student Søren Kanegieser marked the hawkmoth's frontal visual field with black paint so that the animals couldn't actually see in this part of their visual field. They couldn't see their proboscis right in front of them. And tested again, how are the animals interacting with these visual flower patterns? So we had a condition where the eyes were completely free, and then he painted the eyes and tested the animals again. And if you compare where the animals place their proboscis in these conditions between the free and the occluded condition, you already see that in the occluded condition, animals still actually contacted the flower patterns at the edges where the patterns intersect with the rim of the flower. So clearly they could still see the patterns, which is really important. There was still a visual behavior going on. But what you also see is they're not so nicely aligned with the flower pattern anymore. So clearly they don't seem to be probing along this pattern on the search for the nectary. Skip this analysis. And we further confirmed this by actually looking at, once the animals contacted the flower, are they still moving their proboscis along these patterns? And we did that by extracting essentially the orientation of their movement tracks. So this is the proboscis moving across the flower of this animal. And if we take its dominant, essentially direction, or it's actually its vector, its movement vector, then we see if the animal's eyes are free, that their movements are aligned with the flower pattern. But if we occlude just the frontal visual field, the animals are still flying, and you saw that they're still contacting the flower patterns at the edges, but their proboscis movements are, once they're on the flower, are not actually following these patterns anymore, or the pattern direction anymore. Suggesting that the hawkmoths really use these visual flower patterns on our artificial flowers to guide their proboscis on the flower in search for the nectary. And what I forgot to say is on search for the nectary, because in training, we would give the animals sugar water to get them interested into these flowers, even though you don't actually have to. They're naturally interested in this kind of configuration. But in all of these experiments, there was no sugar water on the flowers. So that's also what makes the animals search and search and search, because there's nothing there. So we really get to see how they're looking for a nectary or for nectar on the flower using these visual patterns. All right, so if we sum up what we've seen on the function of these visual guides for bumblebees and for hawkmoths, so very generalized and a much more specialized pollinator, we see that both actually use these visual patterns to orient their approach flights. Both use them to make contact with the flower, once in landing and once in contacting with the proboscis. In our well, I should go one step further. In our experiments, the hawkmoths actually use these patterns to guide their approach to the nectary, the approach of their proboscis, whereas bumblebees did not. And I think this will be a really interesting discussion on why might they not? And there are many, well, as we already heard about, cues that they might also use alternative to these visual patterns. Interestingly, neither the hawkmoths nor the bumblebees actually targeted the center of the flowers where the animals should expect the nectar reward directly. Also not if we gave them radial patterns where kind of the directedness of the patterns might point to the center. So something, or there seems to be a very important reason why the animals start in both cases, mainly contacting either with proboscis or with landing the flowers at the edge. And it simply might have to do with that's the simpler way of interacting with these types of flowers, simpler in the sense of flight control, of landing control, of stabilization of these interaction behaviors. And then finally, we have this interesting result of that flower patterns also guided the bee's departure. And I would like to kind of, as a short outlook, end this overview of what I've shown you about the function of visual nectar guides in these two insect pollinator species with, well, the very obvious, or how do you say, the elephant in the room. In this case, it's a very artificial looking petunia flower that much like this, we've tested all of this behavior of the animals and the function of visual nectar guides with very, very artificial patterns. And patterns in the lab that are very high contrast, very wide, that might or might not have much to do with flower patterns on natural flowers. So a very big gap in our understanding on all of these functions that I've shown you in how they actually play out in natural flower patterns, and something that would really be interesting to look into in the future. And I might just want to give you like a few cues of or clues about what we actually know about the parameter space of what the animals, the bees and the hawkmoths can actually use in terms of features of flower patterns that might help us understand a little bit of how that relates to natural flowers. And that's not even talking about those crazy things like cultivated flowers that are bred for our amusement and that really have very crazy patterns and color combinations. That would be really interesting to see what the animals do with that. But first, just a little outlook on where this might be going and where we might be getting into more questions of what does that actually mean in terms of the function of nectar guides on real flowers. We have also tested in the hawkmoths and in the bumblebees how fine these patterns can actually be for the animals to still use them for their approach guidance, for landing, and in case of the hawkmoths also for guiding their proboscis on the flowers. And I will just give you the very, very short answer on both of them. In millimeters in hawkmoths, they can be 0.25 millimeters fine. So that's actually really, but it got to the edge of what our printers could actually do. Maybe we didn't have the best printers, but they can be really fine, and the animals are still generally aligning their bodies and generally following the outline of the patterns as they're probing. And if we take like the distance a hawkmoth on average has from a flower, that's the width of these patterns is less than a degree of visual space for the animals. So it's really getting down to their resolution threshold of how fine a pattern they can still use. And in a nutshell, this was the same for the bumblebees. So here we tested how the bees aligned their landing on these on stripe patterns that we made finer and finer and finer. And the bottom line is we still saw a lined landing for patterns that were interestingly also 0.25 millimeters wide. Just for context, the next finer category, both for bumblebees and for hawkmoths, was half that, 0.125 millimeters. In the bumblebees, we don't know exactly at what distance they actually make their decision to land. So we have less clearer indication of what that actually means in terms of visual resolution. But certainly they can use very, very fine patterns for these types of behaviors. And a final point, and no data here, but the visual overview. In the bumblebees, we also tested many different types of color combinations with these patterns. The original idea was to use color combinations that would not have any green contrast, not have any color contrast. Sorry, I don't know if you see my pointer. I might be pointing right. I start from the left side, so we make this correctly. On the left side, not have any contrast in the UV channels of the animals, the UV sensitive photoreceptors, not have any contrast for the blue, not have any contrast for the green receptors, and then not have any color contrast, but green contrast, and not have any green contrast relative to the background that we tested the animals on. And the bottom line is, it didn't matter what we showed them, they were capable of aligning their approach flight, using the patterns as landing features, just like they did with our very, very high contrast yellow and blue patterns that we used as controls. So from all we can tell, at least with the color combinations we've tested, the animals can use a very wide range of different color combinations, potentially some that only require chromatic vision and some that only use achromatic vision, as much as we can tell. So this might give a first glimpse into how, well, at least the parameter space that the animals have to deal with when they're actually seeing real flowers. But of course, it really pushes us toward taking the next step and testing what features of actual flower patterns can the animals use for these behaviors within this parameter space of very, very fine patterns that the animals can use, and potentially a large array of color combinations. And so with this overview of the function of visual nectar guides for a generalized and a very specialized insect pollinator, I would like to thank, very importantly, all the people who have been working on the study. I have already highlighted Robin and Alex, who worked on the bumblebee study. I've mentioned Alexa and Nadine, and I should really highlight Sören's contribution. He did a lot of work on understanding how the hawkmoths actually use these visual patterns to guide their proboscis on the flowers. I'd like to thank my collaborators funding that supported these studies. Just a shout out that we are now very recently have moved to Konstanz University, and we will look much more into how flower patterns guide pollinator plant interactions. And so there's still room to grow here if anyone is interested or would like to spread that information. And with this, I'd like to come to an end, and I'm looking forward to discussing and forward to your questions. All right. Shall I close the screen sharing?
If there are some questions that might come up about the talk right now, maybe it might be better to have it.
Okay.
And thank you for the great talk.
Yeah, very nice. Thank you. Do we
I think I had a bit of a naive question about so at the beginning you talked more about the sort of handling of the flowers by the bees. That did seem to be affected by whether there was a pattern or not. So this idea of the approach. And I was kind of curious sort of in terms of, because I think the comparisons that you did were with sort of no pattern versus the sort of a line and then a cross. And is there something to do with the complexity of the pattern? Sorry if I missed this or misunderstood, but because the contrast there seems to be pattern versus no pattern. That could be one way of interpreting it, versus pattern versus specific pattern. Does that make sense? And I was wondering.
Yeah. I mean, I totally see your point, and I think I can only half answer this because we only tried a limited complexity of patterns. So we mainly tried kind of radial versus a very simple stripe, just more bilaterally symmetric pattern, and then and no pattern, and different versions of the radial pattern. So we didn't really try a massive amount of complexity. And for these simple patterns, we saw the animals taking less time to do this entire flower interaction from approach to finding the nectary than they took for when there was no pattern. But yeah, it's hard. And what I can tell you is it didn't, so the animals were equally fast, whether we used this radial pattern extending to the edge versus the radial pattern just in the center, or just the bits of it on the outside of the flower. So that difference in complexity didn't seem to make a difference as long as the animals had these clear indicators. My gut feeling is what actually, or what we saw, what affected the interaction times was really the animals' approach times were shorter when we had patterns, and at least in our experiments, and their departure times were shorter. To me, that indicates that the animals, at least for the approach bit, what they needed was clear cues. Where can we land? That sped up their approach and the landing time they took. And I would think any pattern that gives them reasonable contrast would do that. But I don't know. Yeah, would be an interesting question.
I think we can stop sharing. Yeah.
Yes. I just wanted to ask a very quick related question, just about choosing between different flowers. Like, do you think that they would have a preference for pattern versus no pattern, considering that pattern gives them an idea of where they might be able to land, for example, sort of more certainty or more sort of confidence about what they might be able to do?
Yeah, so we haven't tested this, and I forgot to say it in the beginning, but yes, absolutely. So there's many different insect species have been tested, including, I think, pollinating flies, the bees, different types of bees, different types of lepidopterans, and they all have a preference for certain pattern types, generally over no pattern. And then for specific pattern types like radial patterns, also bullseye patterns, circular patterns, exactly the ones that you also find on flowers. So yeah, but whether that is, and now, I mean, now we're in the discussion, but is that because these patterns help them approach and land? Is that because they have an innate concept of what a flower is and where they find food? Yeah, it's more difficult to answer.
I have a direct follow-up to this, which I think covers I think a question that you, Anna, had for Mike, maybe in any case. So you can both comment a bit on the relevance of these different cues in the context of this whole behavior, right? Because I think that theme has sort of emerged, and I'll just
Well, thank you for asking. This is me, because I mean, from what we did here, especially with the bumblebees, a certain hypothesis emerges, right? That the visual patterns are very important for the animals for the more long-range interactions with the flower. And I think that goes into my question: at what range can the bumblebees actually sense heat, potentially humidity? It does make a lot of sense to me conceptually that visual patterns are very, very beneficial for the more long-range interactions, especially when it comes to landing control, because they give high resolution, very specific information, which you wouldn't potentially get from olfactory cues on where can I land, where is the flower, is this the right flower as well? But then once they landed, they have a ton of other cues. They have the surface cues, they have temperature cues, they're closer, so they have the humidity cues. And they might be relying more strongly on these rather than kind of walking along the visual path that is under them. That's what emerged a little bit from these experiments.
So I can you hear me? Yes. Sorry, still worried about technical difficulties. I would totally agree with that in that the because I was a little bit tight for time at the end, I kind of brushed over the potential explanations as to why you get different outcomes with regards to the combined guide of color and scent versus color and temperature. One of those things might be to do with how temperature works at a very close range. Temperature can really only be perceived by contact almost, or contact with things that are warmed up as a result to it. So the bee isn't able to really make decisions on temperature until it's basically already landed. So that might have that might have something to do with why the speed wasn't greatly affected, because the biggest determinant of speed was, well, the bee is already landing in the right place and is just able to get straight on with it. When with temperature, it needs to do a little bit of a search to find that pattern in the first instance. When it does the right thing. So it's like, with all these different modalities and different cues, certainly something like visual that works very long range and is quite a good consistent cue is probably going to be overall more important. Like I say, these overlooked cues perhaps need to be considered, but I wouldn't turn around and say they're necessarily more important than something like color or factory cues. But the issue is that they do in fact work together and can give that kind of refinement of this overall response we're looking at. So they might be using more information than just these very obvious things. I think that does tie into some of the questions I saw in the comments with regards to when the bees are making these decisions. So with temperature, very much they need to land and then make the subsequent decision. It's not going to affect their landing choices. Humidity, less so. Humidity they can sort of detect before they visit the flower, but it's still quite localized, but like not as like restricted as maybe temperature. But like, so there's a limit to the range at which they can detect these things. So the decision is made quite close to the flower. But if it informs them and tells them, well, we need to abandon this flower now because it's not got rewards, it's not wasting valuable seconds trying to feed on something that's non-rewarding. So it's still useful. So I think that covers a few of the questions.
Yeah, great. Thank you for also keeping up there. Yeah, a little follow-up there. So if we take the perspective of the flower in this, right, and sort of tying in this curious finding that Anna mentioned, the accelerated departure. I mean, basically, I think, Mike, at the very first introduction slide, you mentioned that basically it's flowers trying to lure pollinators in, right? So is it basically of interest for the flower to, like for the plant, to get them in quickly and out quickly as well, because then they keep pollinating? And is there potentially for this, as well as maybe from a guidance cue perspective, an effect that maybe, I don't know if this is generally true, but I would assume that maybe similar flowers may be related, like genetically even related plants sit in a similar space. So it's even true that they would basically get to related plants. I don't know if that's the wrong way to think about plants. I'm not sure, but basically, is there some collective benefit to them going in and out fast and basically visiting a lot of the neighbors?
Well, like if the handling of the flower overall is faster, the bee can visit more flowers in that amount of time. So the fact that they would depart faster after finding a guide, to me, I don't know what everyone else thinks, but to me that looks like the bee understands and is kind of knows how to handle the flower and knows that at this point it might as well go because it's already found the food. So they depart sooner, and that might mean the bees visit more flowers per unit time and it's more efficient, so they waste less energy searching, they get more visits in the limited amount of time. So it's probably good from the plant's perspective to get the bee to visit the way it wants and then to just leave and get on with visiting other plants, because from the bee's perspective that's more efficient, and that will mean that the bee will prefer visiting these flowers. Also, like the longer the bee hangs around on the flower, the more likely it's going to deposit that flower's own pollen on the flower. So it's also good to get them away from a kind of avoiding selfing kind of perspective. That's my guess.
And just to add to this, because I think similar to you, I think the flowers might be giving cues to the pollinators that, okay, or that was our thought, that if you have a clear pattern, the bee goes there, it figured out, okay, now I've been, there's the nectar, I ate it, that's it. Whereas if there's no pattern, they might continue searching for potential spots to feed. On the other hand, I think, I mean, this is also because we created a very artificial situation where we have a flat flower with no other shape cues, again, none of the other hidden cues on where the nectary is. So I wonder if this, I really wonder, I don't want to put too much into this finding, because I really wonder if that would actually be the case with a natural flower that provides so many more cues of you've now been to the nectary and you can leave, or you've collected your pollen and you can leave. Yeah, but then that's a tricky situation we always face when we try to isolate other cues and just focus on the one that we're currently interested in, and we make the situation much more artificial.
Maybe we ask a few, we have a ton more questions from people. So there is one that I think is more specific to Anna from Shiwen. Shiwen says, lovely talk. In the occluded condition where parts of the eyes were painted over, did you observe any behaviors prior to landing that helped them compensate?
Yeah, that's a really interesting question. So what we haven't really looked at, and I think we should, is how long the problem is we don't have a start. So I would like to know, how long did the animals actually take before they start tapping with their proboscis or land on the flower with their proboscis? And do they take longer with the occluded eyes than without the occluded eyes? The challenge we face is they don't really have a starting point. They can freely move around and at some point approach the flower. What it certainly looks like is that they are close to the flower longer before they make a choice to actually contact it with the occluded eyes. But it's not, we haven't quantified it because it's a bit difficult to find a stage to quantify it. What we have seen when we just, and that's really interesting, when we just occlude one eye, the animals will start to move kind of their proboscis. They can't move it much laterally, but the proboscis tends to be more in the visual field of the free eye, and also more on the side of the, because they are symmetrical patterns and they like to mainly probe on the borders. It's more on the side where the free eye is. So in that sense, if they can, at least with one eye occluded and one eye free, they are trying to compensate to actually see what they're doing, if you want. Yes.
All right. So I think we have a, yeah, you got a, I think, a happy question answer. So there were a couple of questions that I think we can maybe bundle on the humidity gradient around the flowers. So sorry, I'm trying, it's like so much going on. I'm trying to, but I think there was something about whether it's constant over the day, and also a question of whether it's correlated with the amount of nectar inside. So basically, is it dynamic or is it very static property?
So with regards to monitoring for humidity, there are basically, prior to my work, 2 papers, one of which was the one I mentioned in Cornell. So there's not a lot of work actually looking at specifically how stable the Q is over the course of the day and with weather conditions and things like that. Our flowers were cut off from the rest of the plant, so they're not linked to hormonal control, which might change things. They weren't like wilting and drying out. They had water supply and things. So, and the time scales for measurements were like several hours. So across that, we did see some change in some of the species, but how much that could be just they're drying out or the actual humidity is taking time to establish, because sometimes we found it went up during the day. It hasn't been explored, but presumably as all these transpirational processes change over the course of the day as stomata close, the plant takes up less water and stuff, we would expect them to change in like the same sorts of ways. There is the consideration, like I said, that the flowers themselves are kind of leaky compared to a lot of other plant tissues, so they might have less control over that. Also, nectar control might contribute to those kind of changes too. Some plants reabsorb nectar, a lot of them don't. With regards to the role of it, with regards to the role of nectar being removed and the relative contribution of transpiration and nectar evaporation, that has been explored a little bit more. So that Cornell study I mentioned earlier in the evening primrose did do a series of experiments where they drained those evening primrose flowers of nectar and looked at what effect that had on humidity. And they found that the humidity did drop when the flowers were drained of nectar, and it was also affected by various blocking off certain parts of the flower to prevent the water vapor contributing to the headspace. So in that species, the humidity level produced is related to floor humidity production. The thing is with several of the species we sampled, the particular one of the ones I raised is California poppy. Not all of them actually produced that much nectar to begin with, but they still produced floor humidity. So it might be more on a species by species basis. It can function as a direct indication of reward state. So that's a direct, honest cue in the kind of parlance of the Von Arx study. We did some experiments looking at the role of transpiration by like basically covering certain plants in anti-transpirant gels. And we found that also decreased the humidity. And we checked among them, the bindweed and the California poppy to see how that changes the humidity. And in California poppy, it drops quite a bit, which would suggest in that species, maybe it's more to do with transpiration and therefore not so honest a cue, but in other species it might be. So it's kind of yes and no. And that's a really interesting thing to look into, is like, how dependent are they on nectar presence, and how that, because a directly honest signal of nectar presence is really useful for the bees, because then they know exactly how much food is in a flower when they visit it. But it might not be the case for all the species that produce floor humidity, unfortunately. So I hope that covers everything.
I have a quick follow-up on that. So is it possible to change the humidity in the headspace of the flower very quickly to see if the flower will try to go back to the previous level? Like, can the flower regulate it, in some feedback dependent way? Or is that not likely?
I mean, like, it could, various plants have various control over nectar production and transpiration. I don't think any of those processes are something that's going to change rapidly. They're quite slow, passive processes. So they might have influence over it, but I don't think it's going to be able to change very rapidly. The relevance is how quickly this cue might re-establish after the bee is visited, if it's functioning as some sort of honest indicator, and how that actually relates to the ward state of the flower, perhaps. But as said, I think it has the capacity to be a directly honest cue, but stuff we've found has found that isn't necessarily always the case. And I don't think there's been a lot of work seeing how adjustable this is, like, generally speaking, with regards to the flower reward state in like lots of species. You really only looked at a handful, so it's quite hard to generalize.
I think there's also a couple of questions. I think one from me and one from Hannah, which are basically talking about the same thing. But in the experiments where you do the cross-modal transfer, you have the hotter part of the pattern corresponding to the black region of the visual pattern. And is it possible that if, bees have some expectation of what a hot visual pattern would look like, they would actually show some transfers? If you reverse the colors, maybe would it be likely that you see something, or do you think that that doesn't matter?
Yeah, so I did see that question was put in the thread, and that is an interesting idea because we were looking at it in terms of the dark parts of the flower tend to be where the temperature pattern is, but if the bee is sort of going on taking a slightly wider view, and that flowers tend to be hotter, it might have this kind of pre-existing association that the hotter flowers are also the bright things. So it might. We did it in a much more kind of, perhaps from the flowers' point of view side, that these two things often correspond, and it's whether they can transfer information learning one of these things against another. So I can't really comment on whether the bee expects bright things to be hotter, but that is a very reasonable expectation because flowers tend to be brighter and tend to be hotter relative to everything else in the bee's environment. So we didn't look at that, but that is a very interesting idea.
I also had oh, sorry. Okay, I also had it's more of a comment or a general question about the generalization experiments, and the fact that bees could transfer from the olfactory patterns to visual patterns, but in your specific experiment couldn't transfer temperature and vision. And what I've been wondering, with the type of experiments that were done for the transfer from olfaction to vision, and this isn't about the specific paper, this is actually something that I think we always have to consider when we talk about patterns. Like, what is I'll go one step back. What is a pattern on a flower, whatever it is, temperature pattern, flower pattern, and what might be more like an inflorescence pattern or a arrangement of flowers? And what I've been wondering with this transfer of olfaction to visual patterns, because it had lots of little holes for the olfaction and then lots of little dots for the flowers, the animals might also perceive that as a flower arrangement. And what they can actually do is a transfer of spatial locations, but what they can't do is a transfer of kind of modalities within a flower. But it's a very difficult question we've also seen responses of both hawkmoths and bees to patterns on one flower that very much looked like they were treating them as individual.
This is something flowers, that is a very interesting way to look at it, and I think with a lot of this sort of floral signaling stuff, it tends to be focused on flowers as an individual unit, but often similar flowers can be clumped together. So like with the humidity responses, I think one of the questions was also on this. It could be that, like we showed that bees can respond to humidity in a flower foraging context, but whether the bees are responding to the humidity at an individual flower's headspace or the headspace of a group of flowers. So like some of the species we sampled didn't produce a lot of humidity on an individual flower unit, but some of them are quite small flowers that might occur in a clump that together make quite a lot of humidity. And in the end, the humidity is the same when the bee is visiting that flower species functionally. Taking this step out and trying to sample groups of flowers and, things like that is something that hasn't been done yet and would be really cool. And it kind of like, maybe they're using these modalities less in a flower associative task and more in a spatial task is also equally possible. And that might be kind of really important to doing this kind of learning. So they're not like learning necessarily an individual flower's contribution to that, but broadly what is going on when they're visiting the flower. And that might be more contextual learning. I don't know. With the humidity, it gets more interesting because like even the vegetative stuff might still produce elevated humidity. So like a forest will have higher humidity than a desert, and the bees might learn to use that at a different scale and things like that. So like it's a very interesting question. And yeah, with the Q transfer stuff, it's possible something very specific is going on that might have more to do with specifically what was done in Lawson's work, maybe. And maybe that's why we didn't find it between temperature and the dark visual patterns, because what we did was not quite able to translate into how bees were able to do this transfer. So I think it's a valid idea.
I think generally it's like, just to come back to your point, I think it's kind of an experimental challenge. How do we read out from the animal's behavior what it's perceiving? Like how it is perceiving the flower? Is it perceiving it, what we show it as one unit, as one flower, or is it perceiving it as an arrangement of flowers? And I don't necessarily think we can just go by size, because as you said, you might have very tiny flowers that together don't take more space than one large flower. And so it's actually, I think, not a trivial problem to figure out from measuring behavior, how are the animals actually perceiving and treating these features we give them.
Yeah, so I think I want to squeeze in like one last question or comment. I think that you both touched on already, but to make this more explicit, I think there's a connection. So Dennis asked, do factors or variables from optimal foraging, like density or frequency of flowers, affect how certain cue modalities are used? And so I think maybe I want to add just a little bit also, like, I think here, from the behavior perspective, right, we're sort of focused on this very close range behavior part. And so, and I think it was mentioned a couple of times, like that maybe how much energy is used during this whole foraging is relevant. So I think what maybe factors in here in this question as well is like how much energy is expended on this like local interaction versus how important are like larger interactions, and is there any evidence for sort of patch like foraging and then sort of jumps between things, and any evidence or anything known about cues that play a role for potentially different stages in this behavioral sequence?
Well, I mean, certainly we've touched on different kinds of cues having different ranges, and certainly a cue that travels from a long distance might be more useful from like it can inform the bee's decision before it's even visited the flower. And like with regards to working on spatial scales and stuff, the bee can simply use visual cues to detect flower location a lot sooner than it can with temperature and texture. It has to actually make contact, which can make things difficult. Like insects don't have as good vision as us, so like that range is perhaps not as good as our visual perception, but like it's still a lot longer than like actually touching the flower. And like ultimately what the bee or the forager wants is rewards per time invested. So it's like it certainly it will probably, I mean, we're going into like areas of like what does the bee attend to and things like that, which can be quite difficult to look into, but it's very interesting. And I would assume that bees would attend to the more useful cues first, but like if all of these things contribute ever so slightly to doing it faster and doing things more accurately, then maybe it wants to attend to everything.
Yeah, I can't. I can't add much substantial information to this because we've been so focused on this close range interaction. But yeah, it's an interesting question. We know close range, what features are attractive to different types of pollinators, but does that kind of add up long range? Are 5 preferred color flowers more attractive than XY? So like at a further range, and I think that goes into these energy landscapes. It's not just the individual interaction with the flower, right? It's also, where do I choose to forage? Is this a good patch because I can actually work through these flowers very fast because they're all, lots of the ones I want, very close to each other, or not? And that being said, I have no idea, but it's an interesting, it's an interesting question. And also then how do these sensory cues kind of sum? Like then we're back to, is it olfaction, vision? Do they linearly just add up and you go to the one that has the most? But I'm just wildly floundering here. I have no idea.
No, but it might explain some of the cases where you saw no difference, that maybe on a longer range, right? Like with I thought the bumblebee pattern spectral content didn't matter in your essay, maybe then it matters because it gives you good contrast once you zoom out, like against green. Like that's a bit similar to the all flowers are hot, so I go to the hot area. I don't know.
It might, though I think in our specific experiment, we were interested in if they can use them for these short range control tasks. And clearly they can, even though we thought they shouldn't be able to use some of them. But yeah, but I agree with you. It could be that the reason why they can use this wide range is because at a further distance, it might be important for them to make decisions about which patch is actually a good one to visit.
This has been very lively, but we have been going for like nearly 2 hours. So I think it's probably a good time to wrap up. And like, I'd like to thank both of our speakers again. It was really, really interesting and like wonderfully complimentary, and everyone who asked questions. There's always the option to catch up if you were late on YouTube and rewatch the talks. And otherwise, I hope we see you for the next episode. All right. Thank you. Bye. Oh my God.